Hey guys, welcome back to another video in my AP Biology or Biology Olympiad prep playlist. In the last video we discussed lipids. Today we're going to be talking about proteins. Alright, let's get started. Proteins are incredibly important to the functioning of our body. They perform a variety of functions like defense, storage, transport, they aid in cellular communication, and so many more things. Proteins are made up of amino acids. The polymers of amino acids are called polypeptides. When a polypeptide is properly folded to a specific structure, or even when there are multiple polypeptides bonded together, that forms a protein. Proteins are a diverse group of macromolecules, but all these proteins are actually only made up of 20 different amino acids. The reason only 20 amino acids can make thousands of proteins is because they're arranged in different combinations. It's like the 26 letters in the English alphabet making up thousands of words. Amino acids are the monomer units of proteins. So let's look at the structure of an amino acid. You have a carbon at the center, bonded to a hydrogen atom at the bottom. To the right, you have a carboxyl group, which again is COOH. And to the left, you have an amine group, which is NH2. Now at the top, you have a chemical group. We use an R to represent this. This chemical group is different for each amino acid. It's this chemical group that makes every amino acid different. Now side chains can be polar, nonpolar, acidic, or even basic. These side chains give amino acids properties like being hydrophilic or hydrophobic. Now let's look at how amino acids bond. As you can see, I have two amino acids at the top over here. So how they bond through dehydration synthesis is the hydroxyl on the first amino acid combines with the hydrogen on the second amino acid. And when they combine, they form a water molecule. Now this water molecule leaves and it creates a bond between the carbon and nitrogen of these two different amino acids. So you end up with a molecule that looks like this. Your backbone is NCC, NCC. You have your carboxyl group to the right, your amine group to the left. Carbon is bonded to a hydrogen and a side chain. Hydrogen and a side chain. Now, like we talked about before, bonds formed through dehydration synthesis have different names depending on the macromolecule that they are formed with. So in carbohydrates, this bond is called a glycosidic linkage. In lipids, it's called an ester bond. In proteins, or between amino acids, it's called a peptide bond. Now, if you're given a molecule and you have to identify if it's a polypeptide, all you have to look for is the backbone NCC, NCC. If the molecule has a backbone of nitrogen, carbon, carbon, and so on, then that means it's a polypeptide. Okay, so we looked at amino acid structure. Now we're going to look at protein structure. Proteins have 4 levels of structure. The primary structure, is the sequence of amino acids in the polypeptide. Each polypeptide has its own specific and unique combination and order of amino acids in it. This is what makes every protein or polypeptide different. The primary structure actually determines the secondary and tertiary structures. Now moving on to the secondary structure. The secondary structure is basically the shape formed by a protein when hydrogen bonds are formed between the amine and carboxyl groups, between COOH and NH2. The hydrogen bonds cause the polypeptide to fold into different shapes. A common shape that is formed is called the alpha helix. This is sort of a spring-like structure. So the hydrogen bonds are formed like this. I'm going to draw yellow lines to indicate that. And it causes the polypeptide to twist and form this spring-like shape. Another shape formed when hydrogen bonds are formed is the beta pleated sheet. In this, polypeptides lie parallel to each other and they're held together by hydrogen bonds. In this case, the polypeptides are called beta sheets. The tertiary structure is the overall shape of the polypeptide. Overall shape of the polypeptide. Now it gains this shape because of the bonds formed between the different side chains of the amino acids. These bonds include hydrogen bonds, ionic bonds, van der Waals interactions, and disulfide bridges. Now, we haven't talked about disulfide bridges yet. This is basically a bond formed between two sulfhydryl groups. A sulfhydryl group is a sulfur bonded to a hydrogen. One common interaction that occurs in tertiary structures is called hydrophobic interactions. This is basically where the hydrophobic side chains on the amino acids cluster to the center of the protein molecule to be away from water. Now, moving on to quaternary structure. Quaternary structure is basically where multiple polypeptides form one single protein. For example, hemoglobin contains 4 polypeptides bonded together. The folding of polypeptides that contributes to a protein structure occurs in something called a chaperonin. A chaperonin is a protein that helps other proteins fold. It's basically like a container. So what happens is the polypeptide in its original form, before it's all folded, goes into the chaperonin and it comes out in its folded shape. Basically what the chaperonin does is it protects the protein from harsh environmental conditions that would prevent it from folding. Now, like I mentioned before, a change in primary structure can lead to changes in secondary and tertiary structures as well. When there's a change in primary structure, the backbone of the molecule changes. And since secondary structure depends on the backbone of the molecule, secondary structure changes as well. A change in primary structure also results in changes in the side chains of the amino acids. And since tertiary structure depends on bonds being formed between the different side chains, tertiary structure changes as well. An example of this would be sickle cell disease. What happens in sickle cell disease is red blood cells are deformed. So they're supposed to be this perfect circle or sphere, but they get deformed into this crescent-like shape. These cells don't fit into the blood vessels, and this leads to the vessels getting clogged. This is actually due to a change in the primary structure of the protein hemoglobin, which is present inside red blood cells. One of the amino acids is substituted with another, and this causes the protein to bond weirdly and form a different shape. And because this protein is present inside the red blood cell, the red blood cell gains a weird shape as well, like this, and leads to the person getting sickle cell disease. Protein structure also depends on the protein's environment. Proteins can only function in a specific environment. Any drastic changes in pH or heat can cause changes in protein structure. When a protein is not in its ideal environment, it denatures. This basically means that the protein unravels. So instead of looking like this, its folded shape, it returns to its original shape, which is just a string of amino acids. That is, it loses its secondary and tertiary structures. For example, when a protein is put in an environment that's too hot, the heat breaks the weak bonds that contribute to the protein's structures, and the protein denatures. And that's all you need to know about proteins. If this video helped, and I hope it did, please leave a like and hit that subscribe button. If you have any questions, feel free to comment them down below. As usual, I have linked a checklist of everything you need to know from this video for review and an image of these notes in the description box. Good luck studying, and bye!